welcome viewers. So far we have discussed about glycolysis, and I'm sure you all are very much aware and familiar to all the steps of glycolysis and all the enzymes of glycolysis, correct? So in the last lecture I told that this is not the end of glycolysis, rather glycolysis is highly regulated, which is known as metabolic regulation of glycolysis, because you need not to catabolize glucose every time. Got it? So when we are discussing the regulation of glycolysis, we need to focus on the three enzymes which made the steps irreversible. And yes, these three enzymes are hexokinase, the first enzyme, phosphofructokinase 1, the third enzyme, and pyruvate kinase, which is the last enzyme of glycolysis. So in this chart you can easily see that there are a few activators of these enzymes and a few inhibitors, which actually which actually make their way, or which actually signals that when these enzymes need to be active and when these need not to be active, correct? So starting from the first enzyme, which is hexokinase, the activator of these enzymes are phosphofructokinase 1, because if phosphofructokinase 1 is active, hexokinase is active, and it is inhibited by the product of the reaction, that is the glucose 6-phosphate. Remember, hexokinase catalyzes the conversion of glucose to glucose 6-phosphate. So the product of the reaction inhibits the higher concentration of the product of this reaction inhibits the first, second and third isozymes of hexokinase, but not to the fourth, which is also a glucokinase, a liver enzyme we have discussed in the previous lecture. Okay. So the most potent regulator of glycolysis, if the question is asked to you, your answer would be phosphofructokinase 1, not the hexokinase 1, because this is the enzyme which is the most potent regulator of glycolysis. And if you remember, I told you that the product of this reaction, fructose 1,6-bisphosphate, is actually the committed step of glycolysis, because when this enzyme is active, formation of fructose 1,6-bisphosphate takes place, and formation of fructose 1,6-bisphosphate actually leads the way clear to further more steps of glycolysis, means it makes sure that yes, glycolysis will go ahead. Okay. So if we talk about the activator of this enzyme, we have seen that it is AMP, and there is one more molecule which we are not aware of it, which is fructose 2,6-bisphosphate. Are you aware of this? No. We have discussed fructose about fructose 1,6-bisphosphate. So keep it in your mind, we are going to discuss in the next slide. And the inhibitors of this are ATP, citrate, which is the initial most intermediate of the citric acid cycle, and yes, phosphoenol pyruvate, the second last product of the glycolysis, inorganic phosphate, and of course hydrogen ion. Why hydrogen ion is an inhibitor of PFK1, we will also discuss. And the third and the last enzyme is pyruvate kinase, and which is activated by fructose 1,6-bisphosphate and inhibited by ATP, long chain fatty acids and alanine. Okay. So this is your fructose 6-phosphate molecule. This is your PFK1, means fructose phosphofructokinase 1. We are familiar with this, and the product catalyzed by this reaction is fructose 1,6-bisphosphate. We know that. Okay. We are familiar with the fructose 6-phosphate, we are familiar with the PFK1, and we are familiar with the product fructose 1,6-bisphosphate. But there is a story behind this also. A reverse reaction is also possible, catalyzing by fructose 1,6-bisphosphatase 1, which we have not studied in glycolysis because this is not the glycolytic enzyme. Why we are discussing this enzyme right here? Because we are discussing metabolic regulation of glycolysis. So, focus on the PFK1, and remember that in the previous slide we have noted down that fructose 2,6-bisphosphate and AMP are the positive regulator of phosphofructokinase 1. This enzyme is very much similar to the structure of hemoglobin. Hemoglobin we have discussed in the protein three-dimensional structure. It is a tetrameric protein having alpha 2 beta 2 tetramer. Okay, means 2 subunits are there and 2 subunits are multiplied by 2. So, the structure of phosphofructokinase 1 is very much similar to the structure of hemoglobin. We are not going into the very much structural details of this, but I just want to aware of this, that the structural similarity between this enzyme and those enzymes which you have previously studied. Okay. So, fructose 2,6-bisphosphate and AMP are the positive regulator of this enzyme, and citrate is the negative regulator of this enzyme. However, citrate positively regulate the reverse step enzyme, and these two moieties have negative impact on the reverse step enzyme. Okay. So, the point I just want to make you sure that you need not to focus on these two positive and inhibitory effects or this fructose 1,6-bisphosphatase enzyme. Rather, you need to think that fructose 2,6-bisphosphate is a potent activator of phosphofructokinase 1. But the question is how this molecule is synthesized. So, the answer lies beneath is there is an enzyme known as phosphofructokinase 2, okay, found in the liver. So, you might be thinking that since it is phosphofructokinase 2, it would be similar to phosphofructokinase 1. No, that is why I have discussed the structure of this enzyme. Okay. Phosphofructokinase 2 is a different enzyme found in the liver which catalyze the conversion of fructose 6-phosphate to fructose 2,6-bisphosphate by adding one more phosphate group at the second position of the fructose, correct? And there is a reverse reaction also happens, catalyzing by the different enzyme known as fructose 2,6-bisphosphatase. Phosphatase means breaking down of phosphate group. Okay. So, how this enzyme cause the positive regulation of phosphofructokinase 1? So, in this slide I just want to make you sure that focus on the high levels of citrate on the negative regulation of PFK1 and high level of AMP and fructose 2,6-phosphate on the positive regulation of PFK1, while high level of citrate causing positive effect on the fructose 1,6-bisphosphate 1 and high level of these two enzymes on the inhibitory effect of fructose 1,6-bisphosphatase 1. You can ignore these two steps because these are the this is the enzyme of gluconeogenesis. Okay. So, let us take a look at the structure of this PFK2 or FBP2. Usually these two enzymes are thought to be similar, or people can take either one or this name, but this is not the correct thing, because as I told in the earlier lecture that in biochemistry if there are two names there are different meanings. So, phosphofructokinase 2 and fructose 2,6-bisphosphatase 2, these are one protein actually having two domains. This is the one protein having two domains. Okay. So, it is a bifunctional enzyme. How does it work? When this protein is phosphorylated, FBP2 is active. The second part of the enzyme, the fructose bisphosphatase 2, is active. And when fructose 2,6-bisphosphatase is active, what will happen? When this enzyme, look at the reaction below here, when this enzyme is active, conversion of fructose 2,6-bisphosphate to fructose 6-phosphate takes place, and if fructose 6-phosphate is present in higher concentration, glycolysis inhibits. Got it? Now let's see when phosphofructokinase 2 is active, and when it is active in the dephosphorylated form. No, please focus on the 2 points written on the bottom of this slide, that if this complex is phosphorylated, FVP2 is active. We have discussed, if FVP2 is active, glycolysis is inhibited, or you can say gluconeogenesis is active. Okay. And if this complex is dephosphorylated, then PFK2 is active, because PFK2 does not need phosphate to be active. It is happy without phosphate. Okay. So, if it is happy without phosphate, it is activated in absence of phosphate. And if this enzyme is active, focus on this reaction, what will happen? The forward reaction takes place, or formation of fructose 2,6-bisphosphate takes place. And we have studied in the previous slide that fructose 2,6-bisphosphate leads to the activation of phosphofructokinase 1. Got it? And phosphofructokinase 1 causes conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Just take a look on the previous slide. Yes, fructose 2,6-bisphosphate causes conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. Means if fructose 2,6-bisphosphate is in high concentration, fructose 1,6-bisphosphate is in high concentration, and if it is in high concentration what will happen? Glycolysis is active. Got it? So, please don't confuse in these two enzymes, because yes, these are the part of the same enzyme, but these work differently. One enzyme, if activated, promotes glycolysis, and the other enzyme, when it activated, promotes gluconeogenesis. Okay, it's simple. So, one more point which you got to know from this slide is that not every enzyme needs to be phosphorylated to remain in active form, and the example is PFK2, because it is happy without phosphate. It doesn't need phosphate to be active. Okay. So, keep these small things in your mind, or note it down in your personal diary. So, coming to the last slide of this, and we will discuss a few questions. We have discussed about the regulation of glycolysis, because there are 3 enzymes, and we have seen how these 3 enzymes are activated and inhibited. So, the question will be in your mind when you have seen the first slide, that why AMP is a potent activator of PFK1 not ADP. PFK1, when you remember this enzyme causes the utilization of ATP, or this enzyme comes in the preparatory phase of the glycolysis, consuming ATP and forming ATP. We have seen, means there are ATP and ADP, not AMP in the reaction catalyzed by this enzyme. However, it is not ADP but AMP, which is a potent activator of PFK1. So, the question will be in your mind, and the answer is, when ATP is utilized rapidly, means when glycolysis is going on at full speed, what happens? And another enzyme comes into action, which is known as adenylate chylase, which causes combination of 2 ADP molecules forming ATP and AMP. Why so? See, when ATP requires in a reaction to produce ATP, then what happens? ATP concentration is going to low, and concentration of ATP is increased. But if glycolysis is occurring at a full speed, you need a continuous supply of ATP. Got it? But if all the ATP converts into ADP, glycolysis will stop or inhibit. So, an enzyme adenylate kinase comes into action, which causes or which takes place the responsibility of the lower concentration of ATP to replenish with a higher concentration of ATP. Got it? So, it is the ATP and it is the AMP right here in the reaction. So, it is not ADP, rather AMP, a potent activator of PFK1. Clear? Second question. What is feed-forward stimulation? So, we know that abundance of fructose 6-phosphate results in higher concentration of fructose 2,6-bisphosphate, because fructose 2,6-bisphosphate leads to higher concentration of fructose 1,6-bisphosphate we have seen. So, binding of fructose 2,6-bisphosphate increases the affinity of phosphofructokinase 1 enzyme for fructose 6-phosphate and diminishes inhibitory effect of ATP. And this is known as the feed-forward stimulation, as glycolysis is stimulated when glucose is abundant. I told you that when there is too much of glucose, body utilize it in catabolism, in glycolysis to give rise to ATP. Okay. So, the higher concentration of fructose 2,6-bisphosphate leads to higher concentration of fructose 1,6-bisphosphate, we know. And if fructose 2,6-bisphosphate concentration increases, it binds to the enzyme PFK1 for fructose 6-phosphate and diminishes the inhibitory effect of ATP, and this is known as ultimately the feed-forward stimulation. Means if you are fed well, then reaction will go in the forward direction. Got it? It's simple. Third, from the first slide, you have also seen that the hydrogen ion concentration plays inhibitory effect on the PFK1. Why so? So, inhibition of phosphofructokinase 1 by high concentration of hydrogen ion means low pH inhibits the excessive formation of lactic acid. Lactic acid formation takes place in actively respiring muscle cells. We will study in the next video when we study Krebs cycle, and when excess formation of lactic acid takes place, the muscle will feel fatigue. So, this is why the PFK1 is inhibited when the concentration, when the pH becomes very low or when formation of lactic acid becomes high. This is a signal that yes, you need to stop because the muscles are feeling fatigue. Got it? So, keep all these regulatory steps in your mind, and in the next video we will study about the next metabolic pathway, that is the Krebs cycle. So, how do you find this video? Please write in the comment section. Happy learning.